Buenas tardes a todos. Mi nombre es Javier Ampudia, soy endocrinólogo del Hospital Clínico de Valencia y desde este momento la sesión va a ser totalmente en inglés. So, thank you for coming. So, and thank you for this session that will be, a session that has been planned to follow one of the initiatives of the European Association for the Study of Diabetes. And as you have seen, the session is Sesión Científica EASD Early Career Academy-SED. So I have the opportunity to share this session with Laura Bruñara. But I want to start very briefly to explain what is the EASD Early Career Academy. The EASD Early Career Academy is an initiative to support, train, mentor, and create opportunities for scientists, clinicians, and students who are at the early and intermediate stage of their career with an interest in diabetes research and/or clinical practice. So its core objective is to provide unique opportunities for developing a platform for a change of knowledge and experience, peer support, and interaction with the WADA EASD community. And we have the opportunity to have here three outstanding scientifics, young scientifics, also who are also part of this board. And, well, together with Laura, we will present all of them. Laura, you want to present the first one?
Okay, so thank you for coming, and thank you all three of you for being here. And so I'm going to present Pierre. Is the first one. No, Pierre. I'm sorry. How do you pronounce your last name?
Larraufie.
Larraufie. I'm sorry. I don't have here in English. I'm sorry. Could you help me with this?
Yes, you can.
Oh, thanks a lot. Pierre is an engineer as a background, but he's working in reproduction and biology development. And he's a researcher in microbiota and gut endocrine physiology at the Institute Université of Paris-Saclay. And we'd like to listen your work. Thank you.
In fact, he speaks Spanish, but I think he will deliver the talk in English.
Muchas gracias por la invitación y la oportunidad de presentar a mi trabajo. No hablo muy bien español, so my presentation will be in English, so thank you very much. Thank you for the introduction. So my talk will be about microbial regulation of incretin hormones. I will focus on all the gut hormones. So I have no conflict of interest to present here. And quickly, you will move. So you are all very familiar now with incretin hormones. Has been quite a big hit in terms of diabetes treatment with GLP-1 agonist or inhibitor, DPP-4 inhibitors, as well as more recently the use of dual or triagonist using several gut hormones. And as you know, there are many other gut hormones which are produced along the intestine with several functions. So along the incretin hormones regulating insulin secretion in high glucose levels, there are also hormones which regulate food intake, bone metabolism, adipose tissue functions, but also immunity. And I think it's very interesting to look at all the hormones at the same time, as we learn from using dual or triagonists, where sometimes it's good to combine the effects of several hormones at the same time. What is interesting about gut hormones is most of them are peptides, which means they come from processing of prohormones. And as you can see, here, they share very similar sequences between families. So that really helps to design drugs which can target or mimic 2 or 3 different agonists. And what is interesting also is because they are all peptides with different maturation, is we can look at them as using mass spectrometry. And this is something I developed as a postdoc when I was in the University of Cambridge in Professor Gribble and Professor Rahman's group. And I was very lucky in my early career of my postdoc to join a team where there was an expert on mass spectrometry, Richard Kay, who just acquired a mass spectrometer, and we said, okay, we know these are peptides, how can we define, rather than using immunoassays, to precisely know which peptide is produced in the gut? And we managed to develop a nice method, which I think is not optimal like all methods, but it works very well, and we're able to detect most gut hormones to also define probable new gut hormones with incretin or regulation of glucose homeostasis. And this is an example of a mapping, of the hormones we could see in different human tissues we collected from different regions we obtained from donors, and we could see that we could detect some hormones in the proximal regions like gastrin or ghrelin, and some more in the distal regions. I will make it work. Here, like GLP-1 or PYY. And it was very interesting to see also the proper processing, like glucagon, which is not at all produced in the gut. Other big interest in my career was the cells which produce these incretins, because they don't come as like that. So they come from a specific population of intestinal epithelial cells called the enteroendocrine cell. What is a bit difficult to study them, and it was a challenge when I started as a PhD student and then during my postdoc, is to get the right tools to have them. And now, thanks to the development of new technologies, we can really manage to understand them in a better way. So this is a single-cell transcriptomic analysis published in 2017, when they looked at all intestinal epithelial cells. And what you can see is the cells which stands out encircled in blue are the enteroendocrine cells. It's less than 0.5% of epithelial cells, so it's a very rare population scattered all along the gut, but very important because it produces more than the 20 hormones, and in reality the gut is the largest endocrine organ in the body. However, what is a bit more challenging is when you look more precisely at these cells, just by subclustering these cells, they exist as very different subpopulations, which can be characterized by the different hormones they produce. But again, this is a much more challenging thing to do when you look at all the cells at the same time, because you only have a low number of endocrine cells in your analysis. So I was very lucky during my postdoc, and then when I came back to France, to Paris, to get the a mouse model to study all endocrine cells at the same time. So it's a NeuroD1 EYFP mouse, which means in the intestine where NeuroD1 is only expressed in endocrine cells, the EYFP, so a fluorescent protein, will be expressed very specifically to sort these cells. So this allowed us to sort endocrine cells, or also to easily look at them by microscopy. And one thing we did during a postdoc with a student I supervised, so it was important for me to get help from students, was to characterize deeply the endocrine cells from the colon. And for that, what was very surprising is, as we expected, we saw a lot of different subpopulations, but we also saw that this subpopulation could be associated with a region where the cells were coming from. So it means we had subpopulation which are coming from the upper from the proximal colon, where we had cells which were more coming from the distal rectum. And these were characterized by, for example, the hormones that are produced, but also the receptors, so the sensitivity of these cells to secrete the hormones. We also saw that some of these cells changed their pattern of hormone or receptor expression as they matured from the bottom of the crypt, where cells come from stem cells, and then they will mature. So we'll have kind of immature L cells producing a lot, for example, of proglucagon, the gene coding for GLP-1, so high GLP-1 levels. And when they move up, they will reduce their production of GLP-1 and rather produce secretin, another important hormone. So that was a very interesting thing to look more into details about what the endocrine cells are precisely. And as I told you, we have very different patterns of hormones along the gut. And if we are interested in GLP-1 as one of the main incretin hormones, you will see that most of the GLP-1 is coming from the distal gut. However, how it's regulated is not very well known. So we know for the proximal cells, it's mostly nutrients and food intake which will regulate the secretion of these hormones. However, in the distal gut, the nutrients are not here, or they are metabolized a lot, mainly by the microbiota. And that's why I decided to look more precisely on how the microbiota can regulate distal enteroendocrine cells. Why? First, because its non-enteroendocrine cells are very sensitive to many microbial compounds. And so this is a figure from a review from the Bucket Lab, for which they highlighted that they express several receptors for short-chain fatty acids, for tryptophan derivative metabolites like indole, for secondary bile acids, but also for TLR ligands, so kind of inflammatory pathway regulating enteroendocrine cells. And all these molecules are able to trigger secretion in hormones, but also to modulate the production of a hormone itself. However, we know that other microbial compounds can also regulate in different ways hormones, like the metabolism of the cells, like butyrate, which is a very important compound for colonocytes. When I came back to France, I first did a short stay in a lab in Paris, in which they were really focused on looking at hydrogen disulfide as a microbial compound, which is produced in very high levels in the gut. And then I took the opportunity of a funding from the EASD, a Rising Star Award, and to supervise 2 master students to establish the role of hydrogen sulfide on enteroendocrine cells using a cell line model on respiration and the impact on secretion. And as you could see here, when we add hydrogen sulfide at high concentration, we have a strong decrease of respiration of the cells, which is the red part here. Whereas when we add the same quantity of hydrogen sulfide, but at smaller doses, just scattered over time, we could increase the respiratory oxygen. And this is due because at high concentration, hydrogen sulfide is inhibitory of respiration in mitochondria, whereas at very low concentration, it's a donor of electron and therefore increase the respiration. And then we looked at what was the effect of this increased or decreased respiration on GLP-1 secretion. And to our surprise, we found that at low concentration of hydrogen sulfide, so increased respiration, we had a slight decrease of GLP-1 secretion. Whereas when we inhibited the respiration in the cells, we had no effect. So this was very surprising. We didn't go further to look at the longer-term effect. This was a very short 15-minute experiment. But it was a first hint saying maybe looking at the metabolism might be important in looking at the functions of the cells producing GLP-1. The next step, linking the gut microbiota with the enteroendocrine cells, is quite a long known story about germ-free animals having very high increased levels of GLP-1. And this is one of the Principes paper in 1989, so 35 years ago, stating that they could see in germ-free mice higher GLP-1, but also other hormones. And then, again, thanks to the funding of EASD and other funding from my institute, I could hire a PhD student, Delphine Polvé, and this is her work, so it's still unpublished data and current work, ongoing work, so it's very, not preliminary data, it's nice data, but we still want to go further into our story at the moment. So to deplete, we used the mouse NeuroD1 CRE-UFP to really look at the enteroendocrine cells. And rather than using germ-free mice, we depleted with a set of 4 different antibiotics. And what you could see, so of course, you had this decreased diversity in antibiotic-treated mice. And, sorry, this one. And we reproduce this very high increase of GLP-1, both at fasted level, but also in response to glucose stimulus, like in an oral glucose tolerance test. We then looked at the phenotypic physiological consequences of these increased gut hormone levels, and we could see that we had a smaller, a lower glycemia at fasted state, but also in response to glucose, and also a delayed intestinal transit, which is linked with the role of GLP-1 as delaying emptying, stomach emptying. However, we didn't see any effect on body weight or food intake in these mice, which might be surprising or not because they have a lot of over-adaptation which happens. So then we decided to know where does this GLP-1 is coming from, because here we are looking at a GLP-1 in circulation. So we just did qPCR of the whole tissue in different regions of the gut, and we also looked at all the other hormones. And what was striking was that GLP-1 was increased in antibiotic-treated mice, in red, only in the cecum and proximal colon, but it was slightly decreased in upper small intestine and in other regions. What was also striking was that we had a very different patterns of hormones depending on the regions and different functionalities, like secretin, which was reduced in the cecum, or other hormones reduced or not moving at all. So it seems that antibiotic had a very specific effect, region-specific effect, which we didn't know, because if it's due to the difference in the region, because of endocrine cells being different subpopulations, or because of the environment, because microbiota is different between cecum, colon, or ileum. Then to look deeper into the endocrine cells characterization, we sorted the endocrine cells and did some transcriptomic analysis of the cells of the ileum, of the cecum, and the colon to have 3 different regions with different effects. We reproduced again that within the ECs, the endocrine cells, we had increased proglucagon, so GLP-1 coding gene, in the cecum, but not in the other regions. And what was striking is we also saw many other genes which are differently expressed. And the most of the genes which are differently expressed came from pathways regulating metabolic regulations, so like oxidative phosphorylation, sucrose metabolism, amino acid metabolism, and some pathways coming from the translational protein production, like ribosome or RNA transport. So all this kind of fits to a specific adaptation of endocrine cells to microbiota colonization, which might regulate a lot, some hormones, but not all, and that's still something we are looking at, is to understand why only some hormones are differently produced or not based on the microbiota. So altogether, I hope I will have convinced that incretin hormones, you know how important they are, but it's also important to look at the cells which produce these hormones, the endocrine cells, and even if they are very difficult to study, I am very lucky now to be in the time where we have new tools to study them. I showed you a mouse model to do that. We have single-cell transcriptomics. We are also developing organoids to specifically look at the function of endocrine cells. And in my interest as an early career researcher, I really wanted to focus on kind of a niche project. I think it's an important message for all the young researchers who want to develop their career, is to think what you could do. And I was lucky to have developed techniques, very good techniques to study kind of a niche. And therefore I said, okay, I should continue on that. And because I joined the lab focused on microbiota, I really created a new interlink link to look further into the role of microbiota with projects then to see how we can develop the microbiota, how we can target it to modulate gut hormone secretion and gut hormone production in the gut. And of course, all this work is based or rely on the work of a lot of people. So my current team in Institut Michaelis, and especially my doctoral candidate, Delphine Polvé, my previous lab where I did my postdoc in Cambridge, and at UMR PSNCR where I did the hydrogen sulfide study, and of course the different groups and the sponsors. And finally, it was quickly presented also the opportunity when you are a young researcher to network, and the EASD Early Career Academy is really made for that. So we hope the thing we are developing will be helpful for any of you who are interested. So you can have a look about it, and that's it. So thank you very much. And if you have any question, I will be happy to take them.
Okay, thank you. Okay. Thank you, Pierre. Anybody do you have somebody question?
Thank you very much, and congratulations for this great work. I was intrigued because I didn't know that region-specific synthesis of the hormones, and I was wondering whether if you have one of these sections removed surgically because of cancer or whatever, that creates a deficiency in some of the hormones. Is that the real situation? And I also, because of the microbiota influence in hormone secretion, I was also wondering, is there a region-specific distribution of microbiota through the tract?
Yeah, so I'll try to answer the first question. There isn't much data about removing some part of the gut and looking precisely at the hormones or dropping or not. I would say the best example I have in mind is bariatric surgery, for which gut hormones have been well studied. And we see that when we bypass the upper small intestine, for example, we completely decrease the levels of more proximal hormones like GIP, for example. And then there's also in mind some specific studies with short bowel syndromes, but in which they have higher levels of GLP-1 and GLP-2 because of the nutrients going further down and therefore stimulating the cells in a very specific way. Regarding the second question about different microbiota along the gut, yeah, it is very different. So most of the active microbiota will be in the proximal colon, cecum in the mice, but we see that we have different, because of different environments, different pH, different oxygen levels, we have different families and species along the gut. So we have some microbiota from the stomach to the rectum, but at different concentrations, very low concentration in the stomach, very high concentration in the distal parts, and also different families and different metabolic activities.
Okay, thank you very much, Pierre. So we move to the next speaker, and the next speaker will be Simón Dreher. Simón Dreher is tissue engineer, and he's a postdoctoral research scientist at the University of Tübingen, Molecular Diabetology Universitätsklinikum Tübingen. And in fact, he's interested in tissue engineering and using tissue engineering for diabetes prevention. And the title of his talk is very provocative. It's From Exercise to Prevent Type 2 Diabetes to Exercise in Vitro. Simón?
Thank you so much. So I want to just add to what Pierre just said. If you find a niche early in your career or something that you did that wasn't really done in a specific field, and then you move into that field and can add that in there, this is always a good strategy to go. As was just mentioned, I do tissue engineering, or I did that in my PhD. We tried to create cartilage from precursor cells. So tissue engineering just means that you try to create a model ex vivo, so in vitro culture, that simulates the tissue of interest. And that is hardly done in diabetes, or especially in exercise, and I'll tell you why in a second. And I only in 2021 moved towards the diabetes field. I started with some exercise intervention studies and then progressed from there. So I have no conflicts of interest to declare. But I hope you all know that exercise is a great tool to prevent the onset of type 2 diabetes or ameliorate its consequences, and that it should always be recommended to patients even with type 1 diabetes. It improves insulin sensitivity and glucose tolerance. However, in all these kind of exercise intervention studies, there are around 20% of people who also fail to improve, for example, insulin sensitivity after exercise interventions. So the goal of our group is to identify what on a molecular level drives these beneficial, or in some times prevents these beneficial effects on a systemic level. And my specific goal is to generate models with human cells ex vivo where we can answer these kinds of questions. And the most important organ when we talk about exercise, because it's the most utilized one, is the skeletal muscle. It's also very important for glycemic control because it takes up 85% of the glucose insulin dependent and takes up glucose insulin independent during exercise. It adapts to exercise and it releases systemic factors called exerkines or myometabokines, even metabolites that can then have systemic effects on other organs like brain, adipose tissue, all throughout the body that are relevant for glycemic control. But it's not really known yet what they do exactly. One of these exerkines that we already researched on is TGF-beta, because it was already associated by our group in 2016. Before I even started, the TGF-beta signaling is high in these so-called low responders, these 20% of people who cannot improve their insulin sensitivity, and it's an important regulator of all sorts of signaling pathways and microRNAs and expressed in the muscle. So we did these exercise intervention studies where we have around up to 40 overweight sedentary participants, 50-50 male and female, and they came in to do 8 weeks of endurance training, 30 minutes on a bike and 30 minutes on a treadmill, 3 times a week, supervised at 80% VO2 peak. And we got them characterized and sampled also for muscle biopsies and adipose tissue biopsies. before they even started the intervention in a rested phase, 60 minutes after the first exercise intervention, 60 minutes after the last exercise intervention, as well as 5 days after in a rested state. So on RNA level, we would call that detraining, but on protein level, it's still the training effect of 8 weeks. So what we did, big study that started early but took a long time, was to characterize the adaptation of the adipose tissue compared to the skeletal muscle of these humans, donor-matched humans that did these exercise interventions, because we wanted to know, is the adaptation sort of similar between skeletal muscle and adipose tissue, or is it not? And on a transcriptomic level, we found that it is completely different. So the highlight is down here. Already to the first acute exercise, so the first 60 minutes lead to an adaptation of the adipose tissue that affects lipid storage, lipogenesis, insulin signaling, but also circadian rhythm. And the adaptation is completely distinct. There's virtually no overlap also after 8 weeks of training in what adapts in the adipose tissue and what adapts in the skeletal muscle. So we looked at all the enzymes that are associated with lipid storage, lipogenesis, and all of those are downregulated by acute exercise already after the first one. This is the light red one, but also after acute exercise, even 8 weeks later. So I mentioned the circadian rhythm, so we tried to look into that, and with the second cohort, where we took several time points, with a collaboration partner, we could establish where the circadian rhythm would be, so where these cells, these genes, these circadian genes would peak during the day in the adipose tissue of overweight humans. And we found out that acute exercise, where it should peak, induces these genes further, and where it should be low, it decreases them significantly. And that's quite interesting, because we know that in the adipose tissue of overweight people, the amplitude of the circadian rhythm, which is the regulator of all kinds of signaling pathways, is blunted. So acute exercise is repeatedly capable of sort of restoring the amplitude of the circadian rhythm. Right. So what we know from the muscle is that it adapts in mitochondrial respiration and oxidative phosphorylation, and we could repeat that as well in this study. So after 8 weeks of training, we have a strong induction also in the rested phase of the mitochondrial respiration in the muscle biopsies from these humans. But in mice, we know that browning exists after exercise in the white adipose tissue. So we thought, hey, maybe we can see that too. But we couldn't at all. So there's also UCP1 and other genes. There's virtually no evidence of browning in response to 8 weeks of exercise in human adipose tissue. Right. So earlier already when I started, we looked at the first data here with this TGF-beta signaling, looking at these low responders and the responders of insulin sensitivity. And we found again that a lot of signaling pathways and genes that are associated with TGF-beta signaling are higher expressed in these low responders, that it can't improve their insulin sensitivity, as well as specific microRNAs that are associated with TGF-beta. So now I come to the models, because I come from tissue engineering and I wondered, hey, can we use this functionally as well? So yes, we can use these muscle biopsies, isolate precursor cells, and then differentiate them in culture after expansion to multinucleated elongated myotubes to do functional studies. And I want to go briefly after that for the sake of time, but of course we then treated them with TGF-beta, saw that these microRNAs are increased and markers of muscle differentiation insulin signaling are decreased. So we then had the luxury in the cells to also be able to genetically modify them and overexpress these microRNAs, only these microRNAs, and that also led to the downregulation of differentiation markers and markers of insulin signaling. So by a combination of in vivo data and functional studies in the cells ex vivo, we were able to identify a TGF-beta microRNA 143/45 axis that leads to the reduction of differentiation and function of muscle cells and insulin signaling and insulin response. So models are great to do functional studies. If you want to ask systemic questions, you had to go to mouse, but especially for exercise and muscle physiology, mouse is sort of kind of far away from human muscle. So there's the human cells that we also used from muscles. They're donor-specific, but their culture method is very non-functional, non-systemic. So my goal is to overcome this and combine the best of both worlds, and that's very important, especially if we ask, can we use exercise-related questions with models ex vivo, so with human myotubes, but in a culture dish. And in 2012, it was already shown that when you do electrical pulse stimulation, you can get these cells to contract. But I don't know if you noticed, but the video already started. So this is not very physiological regarding contraction. So we tried to overcome this, and using my expertise in tissue engineering, what I learned there is that you usually ex vivo need growth factors that are missing in cell culture. So we found out that there is actually a growth factor that is released by moving skeletal muscle in vivo that is not there in cell culture because the cells don't really express it and it's not given. And that's insulin-like growth factor 1. So we did this huge study that we just published now where we treated the cells with insulin-like growth factor during differentiation to improve their physiology. And once the electrical pulse stimulation starts, you will see that these cells now have a far greater range of contraction regarding simulating exercise ex vivo. And of course, this can be quantified. It starts around 5 days of culture, increasing in amplitude and amount of cells. And we can also simulate tetanic contraction, really like what would happen in a muscle cell when you do, for example, weightlifting. Now, we wanted to see what happens in these cells, why are they contracting more, and we found out that it has mostly to do with all kinds of proteins that make up the contractile apparatus that are higher expressed, but also more properly assembled, as you can see with this striated pattern. And we also found that there is a higher degree of oxidative myotubes, more similar to what we have in vivo in the muscle, where there's also a mixture of glycolytic and oxidative myotubes. So then we went ahead to do Ouroboros measurements to measure mitochondrial respiration, and indeed, I'll make it short, the red line is the new differentiation protocol, and indeed, there's higher mitochondrial respiration. Finally, a very important step is glucose uptake. I mentioned 85% of the glucose is taken up insulin-dependent in the muscle. But in vitro, with the old differentiation protocol in blue, GLUT4, so the insulin-dependent glucose transporter, wasn't expressed. So with the new differentiation protocol, we almost thought it's too good to be true, but it's actually true that we have now more GLUT4 and more insulin-dependent glucose uptake if we stimulate with insulin. So we just now, brand new, published this year, the new standard for 2D human myotube culture that can also answer exercise-related questions. However, 2D is still not very physiological. So regarding tissue engineering, we had early concepts already. How can we get these cells in a three-dimensional shape to have actual muscle fibers? And these are concepts that I drew up with a collaboration partner of molds that then actually made it happen to real cell culture. So in these molds, we cast our myotubes in a collagen scaffold with a frame around them with our new differentiation protocol. And now, they not only individual cells contract, but in this 3D culture, they sort of all pull in one direction because they are aligned by the collagen structure. So we have real sort of linear macroscopic contraction, and we further improved the gel, and now we have this really convincing nice, also frame-moving contraction in 3 dimensions, where we can also do different modalities of electrical pulse stimulation to simulate different kinds of exercise. Now, the final step, of course, is answering systemic questions outside of a mouse. So we're currently finalizing a DFG grant to even further improve this, but we started already with a exercise on a chip concept, where we want to use this new differentiation protocol in a perfused system to add further organs on a chip, where we can also, for example, for adipose tissue, actually use our biopsies from the exercise intervention studies that we did early on, to have donor-matched, again, muscle and adipose tissue to answer questions like, what does TGF-beta signaling or other exokines induce after exercise in the tissues of the same organs, of the same donors in the circulatory system, and excluding hormonal regulation, for example. Now we have early results. This is a 3-layer chip design where you have a cell chamber, a semi-permeable membrane, and a perfusable medium chamber. 2 of them are always on 1 microscopic slide. These are actually from a hard muscle-based chip, so they're actually too small for skeletal muscle. Because it's a different type of physiology, but we were able to seed our cells over time in this culture and also culture them over 10 days, and that's what it looks like macroscopically. So in the cell culture, it's actually a very small chip, but in there, our collaboration partner showed that also only with our new differentiation protocol, you get long multinucleated myotubes, even though the chip is not designed for skeletal muscle, and only with the new differentiation protocol, they contract, so it nicely fits to what we found. Now we're working on a better version of this chip for skeletal muscle. So we have now exercise ex vivo models in 2D, easy to use for everyone basically. Then we have the 3D model where we have actually linear macroscopic contractions of these human myotubes, and we're working on an exercise on a chip model where we can eventually connect other organ systems to answer systemic questions. Now, I want to thank my group, of course, and this work is partially funded by the DDG, and of course the EASD is a great network to help collaborate on all kinds of things. Thank you. And I'm happy to take all kinds of questions, of course.
Thank you, Simon. The communication is open for any questions from the audience. Yeah. Professor De Leiva.
Well, excellent presentation. It's a very interesting model to investigate the effects of exercise. I'm just questioning if you have investigated some appetite inhibitors that are related with a very strenuous exercise, like, for example, lactoyl phenylalanine that have been recently, very recently investigated, I think in your own university in Tübingen as well.
Our group, yeah.
Yeah,
that's
exactly.
part of the DFG grant, very well.
Okay.
Yes.
What is your opinion about the future of
Yes.
this kind of mediator?
It's exactly what we're doing, right on point. So these myometabokines, this is also a term that was keyed in our group, which are metabolites like the 3-PLA and the LAC-FE that are associated to, they are increased during exercise. We saw that also in our intervention studies. But what is interesting for both of those, it is suggested due to the arterial venous measurements that we did, that it comes from the muscle, but it's not completely confirmed. And this is also something that we can do with this model, because we only have skeletal muscle cells in there, so we have finally a way to confirm that these so-called myometabokines are actually released by contracting muscle. And then, of course, in a donor mesh system, excluding hormonal regulation, we will be able to connect adipose tissue, for example, and then see what these myometabokines do with other organs. Because the two you mentioned are associated with weight reduction and fat reduction. Perfect.
Thank you. Thank you.
Okay, Simon, we need to move to the next speaker. Okay. Laura.
Okay. So I'm going to go Domenico Trico. Domenico is assistant professor of internal medicine at University of Pisa. And his research focus has been evaluating the effects of macronutrients on beta cell function and pathogenic role in type 2 diabetes development and progression. Thank you, Domenico.
Okay. Thank you for your very kind introduction. And first of all, I'd like to say that I'm truly honored to stand here today as a representative of the EASD Academy, which I'd like to show you mostly because I want to point out that it's not just made by straight white males. I can say we have become the minority of the EASD Academy, so we really think that inclusion is one of the most important part of our mission. So during the next, I think, 15 minutes of this talk, I'm gonna give you a brief overview of my personal research journey in the identification and formal characterization of nosological entities we call primary insulin hypersecretion, which is basically insulin hypersecretion more than compensating for the degree of insulin resistance. And I will also give you some insights on some proposed lipid-related pathogenetic determinants of this condition. So the story I'm going to tell you starts at the very beginning of the natural history of type 2 diabetes, which we all know is characterized by insulin resistance and a parallel increase in insulin secretion. Now, the traditional view of insulin hypersecretion in this early phase of the disease is that it's just a mere compensatory mechanism against the insulin resistance to avoid dysglycemia. However, we know today that the relationship between insulin resistance and insulin hypersecretion is actually bidirectional. And there has been, for a few decades now, an alternative model that postulates that, at least in some individuals, beta cell hypersecretion is, in fact, the very first event leading to chronic hyperinsulinemia, insulin resistance, and eventually fat accrual and type 2 diabetes. And so we decided to provide support to this model by identifying a condition of enhanced oral glucose-stimulated insulin secretion, more than compensating for the degree of whole-body insulin resistance measured by the gold standard technique of the euglycemic clamp. And by design, this condition was associated with enhanced model-derived parameters of beta cell function. Here I'm showing the beta cell glucose sensitivity, and in the absence of substantial differences between groups in insulin sensitivity. So, and what we found intriguing is that this condition is associated with worse rather than the better glucose control, and even more importantly, with a much higher risk to develop IGT or type 2 diabetes, even over a relatively short follow-up period. And these findings so far have been replicated in 3 different cohorts of non-diabetic adults and also children and adolescents. And so with this preliminary study, we said that insulin hypersecretion, independent of insulin resistance, can actually directly contribute to different mechanisms involved in the pathogenesis of type 2 diabetes. And so we were wondering which are the pathogenic drivers of insulin hypersecretions. So in other words, which are the biological signals that can generate and sustain an increased insulin release in the absence of substantial dysglycemia. And so we and also other groups think that chronic overfeeding can actually play a pivotal role in insulin hypersecretion by a number of mechanisms, including an increased availability of non-glucose substrates, such as triglycerides, free fatty acids, and amino acids. And in fact, using a targeted and untargeted metabolomic approach in the same cohorts shown before, we found that primary insulin hypersecretion was associated with higher levels of plasma amino acids, free fatty acids, and triglycerides. Now, the biological rationale in support of the role of free fatty acids and amino acids in stimulating insulin secretion is supported by a large body of evidence. While the less robust is the evidence in support of a pathological role of hypertriglyceridemia in insulin secretion and the other pathogenetic determinants of glucose intolerance. So in other words, it wasn't known back in 2017 when we reviewed the literature whether hypertriglyceridemia was just a mere reflection of the overall metabolic status or whether it could actually play a role in the pathogenesis of diabetes. And so this was my data-driven hypothesis back in 2017, a few years ago, which of course I wanted to test more mechanistically. But at that time I was also at the main crossroad of my professional and I'd like to say personal life, having just finished my residency and having a few of quite good career paths to choose between, perhaps being the academy, the most exciting but also the most challenging one. And this is when I heard and I went to joined the EASD Future Leader Mentorship Program for clinical diabetologists, which perfectly suited my needs at that time with its main aim to develop recipients into leaders in the field of diabetes and facilitating their academic appointment. And this program, slightly changed, is still ongoing and there will be an open call soon. So please read our newsletters because you will read the news about this program. So this mentorship program actually gave me the tools and the collaborations to test my hypothesis, again, that high plasma triglycerides can have a pathogenetic role in the early alterations of insulin levels and action that can lead to type 2 diabetes. And to test this hypothesis, I used 3 different experimental approaches: observational studies, in vitro studies, and also a clinical study. So first, in 2 observational studies, again, in non-diabetic adults and adolescents, I found that increasing plasma triglyceride levels over time are associated with higher increasing insulin secretion, both at fasting and after a glucose challenge, and this remained true also after multiple adjustments for potential confounders. So second, I flew to the King's College of London at Professor Peter John's lab, where I carried out a series of static and dynamic insulin secretion experiments in human pancreatic islets and murine pseudo-islets that were exposed to high physiological levels of triglyceride-rich, very low-density lipoproteins. And I found that exposure to VLDL immediately increased insulin release from human pancreatic islets, both at basal glucose concentrations and also at stimulated glucose concentration, and the same was also true in other experiments. So these findings set the foundation for the third study, which was a randomized control trial in 12 healthy lean volunteers who underwent 2 oral glucose tolerance tests with 2 stable glucose isotopes to measure glucose kinetics, and that were run during a 5-hour infusion of either 20% intralipid, a lipid emulsion commercially available, or just normal saline. And we found that the lipid infusion increased triglyceride levels towards high physiological levels with just mild effects on free fatty acids and without affecting plasma glucose and insulin. After the glucose ingestion, however, the lipid infusion markedly deteriorated glucose tolerance, and this happened despite the much higher insulin levels, again during the lipid infusion, and this clearly identifies a lipid-induced insulin resistance condition. And we were interested to see which were the mechanisms for the lipid-induced hyperinsulinemia, and therefore we used C-peptide deconvolution, and we found out that the lipid infusion reduced plasma insulin clearance, meaning that more of the secreted insulin could actually reach and remain into the systemic circulation, and also that the lipid infusion increased glucose-stimulated insulin secretion. So both mechanisms contribute to increasing plasma insulin levels. So these are other novel insights that I couldn't show you for the sake of time, clearly demonstrated that mild acute hypertriglyceridemia could have marked negative effects on glucose metabolism, contributing to the development of chronic hyperinsulinemia, which, as I said, is not good for your metabolic health. So at this point, you should be convinced enough that chronic overfeeding can increase insulin secretion by providing great amounts of lipid substrates. But what is noteworthy, at least to me, is that insulin hypersecretion and the resulting chronic hyperinsulinemia in turn can enhance fat accrual, particularly in ectopic regions, and can alter the normal adipocyte morphology and function in what it's sometimes called the adipo-insular axis. So this is why I carried out my latest study with the main aims to examine whether this new condition, primary insulin hypersecretion, is linked to early alterations in the adipose tissue phenotype and function, And I also wanted to examine whether this complex adipoinsular interplay could influence the trajectories of glucose tolerance, beta cell function, and adiposity over time. And to do that, I analyzed cross-sectional and longitudinal data from this very well-characterized cohort of children and adolescents with overweight of obesity, representing the three most represented ethnic and racial groups in the United States. And all the subjects underwent an glucose tolerance test, the DEXA scan, an abdominal MRI, a biopsy of the abdominal subcutaneous adipose tissue, or SAT, a neoglycemic hyperinsulinemic clamp, and some of them also underwent a long-term labeling protocol with deuterated water to assess lipid dynamics and also follow-up assessments. So very quickly, what did we find? So we found that insulin hypersecretors, classified, as I said before, had increased visceral adipose tissue compared with normal secretors, despite having the same overall degree of adiposity and the same absolute fat mass. They also had increased hepatic fat fraction with a threefold higher risk of having metabolic disease-associated steatotic liver disease. They also have a more hypertrophic adipocyte phenotype, which consists in a right shift in the size distribution profile of cells deriving from the abdominal SAT biopsies, meaning that people with primary insulin hypersecretion had higher adipocyte peak and nodule diameters and the lower absolute number of adipocytes. We know that an hypertrophic adipocyte phenotype is not metabolically favorable. We also found that these subjects had higher leptin and free fatty acid levels normalized for unit of fat mass, meaning that their adipose tissue can have some alterations in the endocrine and metabolic functions, and also that these subjects with insulin hypersecretion had a faster lipid turnover, which depends on not on de novo lipogenesis, but on free fatty acids coming from the diet or maybe from the liver. And this is what this graph represents. More importantly, when we looked at longitudinal data over a very short follow-up time, it's just 2 years, we confirmed that people with primary insulin hypersecretion had a much higher risk to progress towards impaired glucose tolerance, so type 2 diabetes, and also hypersecretors tend to maintain stable their already quite substantial degree of adiposity, while those with normal secretors tend to have a decrease in their overall adiposity. And we were also interested to see whether, in turn, early alterations in adipocyte function can alter the trajectory of beta cell glucose sensitivity or beta cell function. And eventually we found that those subjects with more hypertrophic adipocytes or higher leptin levels had increased beta cell glucose sensitivity at follow-up, while those with smaller adipocytes or lower leptin levels tended to have lower beta cell glucose sensitivity, so a reduction over time in beta cell function. And I think this finding in particular is very important because we demonstrated very well that in this specific population of obese adolescents, the beta cell function tend to decrease almost inevitably during the pubertal transition. So altogether, I think these findings are consistent with the original hypothesis of a complex adipoinsular axis, mutually influencing the trajectories of glucose tolerance, fat accrual, and beta cell function. Was earliest alterations can increase the risk to progress towards type 2 diabetes, at least in populations at risk. So finally, I'd like to acknowledge the huge contribution of many people who worked on these research projects, our funding agencies, in particular the EASD, from which I got 3 grants so far. And of course, I'd like to thank you all for your very kind attention. And please, I will be available now or later on for any question you may have. Thank you.
Thank you, Domenico. We have time maybe for one question, so I think there is one question over there.
Let me say, first of all, thank you very much, all the speakers, and please let me say that all your work is extremely valuable and appreciated, independently of the limited audience today. And regarding your talk, Dr. Trico, let me ask you, maybe 60 years ago when experts were debating about how to diagnose diabetes, and some of them bet for adding something more than only glycemic values for diagnosing diabetes. For clinicians and researchers, clinical researchers, do you think that we should add some fat metabolism for diagnosis, or at least to better qualify or subtyping type 2 diabetes in our clinical diagnosis?
Thank you for the very, very interesting question. Someone has said that if the early investigators of diabetes didn't have the taste for the sugar, we will now be talking of diabetes like as a disease of lipid metabolism instead of glucose. But since the urine tested of glucose, so now we are all talking about glucose. This is, of course, an exaggeration, but I really think that there is a strict interconnection between the lipid metabolism and glucose metabolism. So we may want to integrate some easy lipid-related marker in our clinical workup. I mean, there are already several indexes of insulin resistance who integrate triglycerides, for instance, triglycerides per glucose levels, or triglycerides over HDL levels, which are all markers of an abnormal glucose tolerance, glucose metabolism, and also an increased cardiovascular disease, because we should remember, of course, that type 2 diabetes, people with type 2 diabetes eventually die for cardiovascular disease. So knowing whether the lipid metabolism is altered or not may be also important to define their cardiovascular risk. So I really think your point is well taken. So thank you again for your question.
Thank you. We don't have any time for more questions. So I really appreciate the speakers for attending the time. And also, I think this is a good example how scientific societies should support also young scientists. As you can see there, I think it was a very good session, so with high quality research. And I thank you also, my co-chair, to chairing the session. Thank you very much.